Hello everyone. Today I'll be talking to you about some fundamental neural mechanisms that need to be understood in order to grasp how neurons and neural circuits function. So all neurons possess the potential for electrical activation. This is governed by the existence of an electrical charge relative to the surrounds of the neuron. The cell membrane is a semi-permeable layer that only allows charged particles, or ions, to cross the membrane via channels and transporters. There are specific gradients exist in all neurons, whereby sodium ions, shown here as Na+ particles, and chloride ions, shown as Cl- particles, exist in high concentrations in the extracellular space, and potassium ions, shown as K+ particles, exist in high concentrations in the intracellular space. So this arrangement creates the potential for these ions to move across the membrane via their specific ion channels down their concentration gradients in order to essentially establish an equilibrium across the membrane. So for instance, here is this pink channel here is a potassium channel, and you can see that when activated, a pore opens that allows potassium ions to travel across the membrane into the extracellular space. So this arrangement of ions is established by a series of transporters, and these transporters use energy in the form of ATP to build these high concentrations. In doing so, they also create an electrical gradient across the cell membrane. There are more positive ions in the extracellular space, so it is relatively more positive to the intracellular space. And this negative intracellular environment is what's referred to as a resting membrane potential. So this leads us to what happens when the charge inside the cell changes. So small changes of the intracellular environment, for instance, if it becomes slightly more positive, lead to these failed initiations. This is an increase in intracellular charge that is insufficient to reach threshold. Every neuron has a threshold at which an action potential will be triggered, and any changes that fall below threshold will not trigger action potentials. However, once an action potential is triggered, we see a rapid depolarization. This occurs when sodium channels open, allowing sodium ions, which are in a high concentration in the extracellular space, to flow into the cell, thereby making the neuron more positive. It therefore loses its polarity and becomes more positive too. So this depolarization occurs for a very short period of time, at which this action potential reaches its peak positive charge, roughly plus 40 millivolts, at which time we see sodium channels close. So these sodium channels, while rapid to open, are also very rapid to inactivate. And so this triggers a slow repolarization of the cell. So coinciding with the sodium channels inactivating, we see potassium channels opening. These were triggered at the same time as sodium channels, but they take a little bit longer. And so after a millisecond or two, we see potassium channels open, and you see potassium, which you'll remember is in a high concentration inside the cell. Potassium ions flow out. This makes the cell more negative, and this is referred to as a repolarization. So this repolarization continues until the membrane potential becomes more negative than the resting potential of the cell. And this is what's referred to as a refractory period. And so this refractory period occurs when potassium channels begin to close, and over time, through a few milliseconds of refractory period, sodium channels reactivate and gain the ability to open again. And so during this refractory period, no action potentials or no other action potentials can be triggered. This is for two reasons. The first being that the membrane potential now is much further from threshold, and so it's very difficult to get this membrane potential positive enough to reach threshold. And the second reason is that during most of the refractory period, sodium channels remain inactive. They require a prolonged period of the refractory period to gain the ability to open again. And so eventually, over time, this refractory period levels out and returns to resting state. So one characteristic of an action potential is that they're dynamic. They don't just fire an electrical activation at that site. They move along an axon in one direction only. And so how does this occur? I'll give you a couple of seconds to think try to think about what would happen in the intracellular space, especially during this rapid influx of positive ions, the rapid influx of potassium ions. What would happen to the ions that are already in the intracellular space? I'll give you a couple of seconds. Okay, so hopefully one thing that you thought of is that when we trigger an action potential and trigger a local environment to become more positive inside the cell, hopefully everyone realize that there are already positive ions in that space. Remember that when we get this rapid influx of sodium ions, there is already a higher concentration of potassium ions inside the cell. And so what do positive ions do when two other positive ions? They repel. And so what we would see is this rapid influx of sodium ions would lead to a repulsion of potassium ions along the axon. And this would therefore trigger this neighboring region to become more positive. And so when that becomes more positive, we reach threshold and we trigger another action potential. And this occurs in both unmyelinated axons, as shown at the top, and myelinated axons, shown at the bottom. So the difference being, a myelinated axon simply has these myelin sheaths that wrap around the axon, and they move ion channels away from the sheath. And so you get these spots between ion channels, between myelin sheaths, where all of the ion channels congregate, and these spaces are called nodes of Ranvier. And so in myelinated axons, because there are a lot more channels densely together in these nodes of Ranvier, when we get larger positive influx there, we get greater repulsion force, and positive ions are pushed further along the axon, and they essentially jump to the next node where it triggers another action potential. And so one thing that this shows you is how a myelinated axon can conduct electrical stimulation much faster than an unmyelinated axon, because it's essentially jumping from node to node rapidly, instead of the unmyelinated axon where you just get a local change occurring. However, both of these are very fast in conducting. And so we see these action potentials travel along the axon, and the reason that they're unidirectional is because when we see these positive ions repelled, they're actually repelled in both directions. However, you'll remember that an area that has just been through an action potential remains in its refractory period and can no longer fire. So the action potential jumps to an area that hasn't fired a potential, fires one, and then that continues to jump to the next node. And so it can never travel backwards because of the preceding region of the axon remaining in its refractory period. So these action potentials travel along an axon until they reach the axon terminal. And so at this point, the positive charge reaches the terminal and triggers calcium influx instead of sodium influx. So these calcium ions at the nerve terminal flow into the cell and they bind onto synaptic vesicles. These are small pockets within the axon terminal that contain neurotransmitter particles. And so the calcium ions, once bound to these synaptic vesicles, also bind the synaptic membrane and allow them to fuse together and release these neurotransmitters into the synaptic cleft. And so what this means is that we've now triggered a neurotransmitter to signal the next cell. So these neurotransmitters bind to receptors on the next cell and are able to then trigger action potentials in the following cell. And so these neurotransmitters can be any of the numerous transmitters that are present throughout the brain. So the two major neurotransmitters that you'll have to know initially are glutamate and GABA. Glutamate is the most prevalent excitatory transmitter in the brain, and GABA is the most prevalent inhibitory neurotransmitter. So here is a little slide that summarizes both glutamate and GABA, as well as several other major neurotransmitters, and describes some of their functions in the brain. For instance, we can see here that glutamate is involved in learning and memory and neural plasticity. And while we're on this topic, there is an article posted in the module page that you'll see by Soderberg and Kerlach, and this describes the role of the two main glutamate receptors, these NMDA and AMPA receptors, and what role they have in neural plasticity. And you'll all have to read through this article in order to answer the quiz questions. But over the next week or two, we'll be going through these glutamate and GABA neurotransmitters and their functional roles. And then throughout the semester, we'll cover the various other major neurotransmitters that are summarized here and discuss the role that they play in each of these functions. So I look forward to going through these various functions and neurotransmitters with you throughout the semester. Thank you.